Good morning, good afternoon, everyone. My name is Adrien Motu, and I'm part of the scientific engagement team. And today I'm happy to present you the databases and how you can use those different databases to better select your models for your studies. So those databases I will present, all of that is accessible. You just need to register on it using a password on your mail address. So you can find it directly on our website, or you can directly contact us, and we will send you a link for registration. So as you can see here, we are on the first part of the database called UBase. This part of the database that we have is dedicated to PDX models. As you can see on the left-hand side of the screen, we have different parts for the database. I will start there. This is all the different models that we have available. So the U prime corresponds to the PDX models that we have. You can see that we have more than 2,000 of them across more than 30 indications. So we have more or less models depending on the indication, but all of them can be searched here. Of course, we have different filters that you can use if you want to reduce this list. So we have here first a search function in which you can type the model of interest, for example, if you already know which model you are interested in. And then we have a couple of filters that can be used to reduce the list that you have here. For some of the models that we have available, we have the standard of care we have tested in the PDX. So you can only reduce the list to the models in which we have tested some specific standard of care. There is also the models, for example, in which we have tested the orthotopic and validated the orthotopic implantation. So that can be checked here. You can see we have 27 models for which we have tested the orthotopic implantation. The growth curves as well. I will present this just after, but you can see here on the left that we have examples of tumor growth curves. So you can reduce the list to the model only for which we have the tumor growth curves available. The models we have in a tumor microarray as well, or pre-treated PDX. So this filter is for models for which we have treatment history of the patient, and we know with which treatment the patient has received. On those filters here, you can also select drugs. Here we have the standard of care data only, but if you are interested in a specific standard of care that we might have tested in specific PDX, you can select one on the list and see which models we have tested with that specific treatment. Here we have a lot of drugs already. If you're interested in any specific kind of treatment or drug therapies, please feel free to reach out to us. Even if you don't find the drug here in the list, we might be able to share more information with you in Excel files. Then you also have a filter here for the origin of the PDX, so Asian or Western origins. We have PDX from both origins. And then we have ground bio site availability. As you might know, we have sites across the globe. So in the US, in the UK, in China as well, in which we can perform PDX studies. So all of that is there. If you want us to generate some comprehensive list of PDX available on specific sites, we will discuss that in more detail. So I'm not sure if you will use that filter a lot, but it exists, so it's good to know. And then you can use some keywords and subtypes if you want to look at specific PDX subtypes there. Just to present you the other part here, the really interesting part about which information we have for each PDX, I will use the filters to select one interesting model here. For example, we can look at models that have been pre-treated, so for which we have the treatment history of the patient. You can reduce a list of more than 2,000 PDX to a list of more than 300 PDX still. And then, for example, maybe I'm interested in colorectal cancer, and I will present you one specific model that is really interesting because we have just recently acquired this model here from patient that has been treated with KRAS G12C inhibitor. So it will allow me to present you which kind of information we have for the PDX. As you can see in the summary part, we have the name of the PDX, the number. We have the cancer type then over there. We have the subtype, we have the origin of the PDX. Sometimes we have some papers referencing if the PDX have been used in some paper, in some experiments and it has been published, we have the reference here that can be clicked. For that one, since it's pretty new in our collection, we still don't have reference. And it's also not in tumor microarray, but we have a lot of models that are in tissue microarray that can be used to, for example, screen in a specific indication a target for protein expression before running and selecting the right model to perform an efficacy study against this specific target. Then we have the special features, and for this specific model, you can see that we have a model that is KRAS G12C mutated. It is said that this particular samples from the patient has been taken when the patient was still responsive to KRAS inhibitor. It says that it's an inhibitor responder. And then another very interesting information is that we have other samples from the same patient at different stage of the treatment, and I will come to that a bit later. So there is this kind of information. Sometimes for some PDX here, it's written cachexia. It means that with that PDX, we have observed that sometimes we have some body weight loss. For example, if there is some interaction with the model, it will be also stated here, so you know if a model is more willing to develop some iteration, for example. Then we have more information at the bottom here in this section. You have the gender of the patient, the age of the patient from which the PDX has been taken, the stage of the disease, the grade of the disease if available. We have the pathology diagnosis over there, which corresponds to the pathology diagnosis at the time when we collected the original tissue to generate the PDX. The biopsy site as well. Here it's coming from abdominal wall. Treatment history. Here we have we know that this patient has received in first and second line some follow-outs and FOL1R1. And there is a third investigational line of treatment, and we know that it has been KRAS G12C inhibitors there, and that the patient has responded to this treatment at this stage of the disease. In addition to that, we have some pathology QC. So pathology QC corresponds to a control that we have on the PDX. Here you see it has been done at cassette number 4. And then we might have some more information on comments here, if available. Here for that new model, we don't have a lot, but you can see here, and I will present that later, that we have RNA-seq data, exome sequencing data, whole genome sequencing data for the model, and we have that for almost all the models in our collection. And you can have a look at that by yourself and look at specific gene expression, mutations, copy numbers as well for a lot on, if not all models in our collection. I will present you this part of the database a bit later. And then the final information that we have is the mouse strain in which we have validated the PDX, the site availability. You also have the virus status of the PDX over there. You can see it's mostly negative for all the virus that has been tested. You have the HLA type of the PDX here. You can click and you have the HLA A, B, C, and the source, the method that has been used to generate those HLA types. And then we have the genomic feature. Genomic feature is interesting because you have a list of specific mutations and gene amplification or deletion, specific deletion that might be interesting for. So it can be interesting to have a look. But if you are interested in more specific mutation, you can also check that with the other part of the database I will present just after. And then at the bottom, you have some examples of tumor growth. First, you have this part there where you have the body weight curves. It can allow you to see, for example, if there is how severe a cachexia here is. For example, here in the spatial features, you can see that this model has been listed as potentially cachexic. And you can see here that there's slight body weight loss when you grow the model. And then when you click over there, you have an example of how the model is growing without any treatment. With some other models we have used a lot more that are a bit older in our collection, we have a lot more examples, and you can see how heterogeneous is a model in between different experiments, and that can be very valuable data to have. And at the bottom here, you have some example of standard of care. As you might remember, this model has been taken from a patient that has been treated with KRAS G12C inhibitor, and what we have done is we have tested that on the PDX we have generated from the original tumor tissue we have taken from the patient. And you can see here when we test some G12C inhibitors like sotorasib or adagrasib, we see a good response of the treatment and a good tumor growth inhibition with both G12C inhibitors. So it validates the fact that this patient was sensitive to the G12C inhibitors, and that's a model that we can use. We have the treatment history. We know the patient was responsive to the treatment, and we also know that when we test it in the PDX, we also see the same kind of response. Now I will go to another models, and I will select maybe one of the models, PDX models that has been generated from the same patients. So we have two of them, so I will take one. I will open a new window here, so we can compare the standard of care response, because it is something that might be interesting. So I will use 9547 model from the same patient. I will go up again. And there you can find again the same information. And you can see in the spatial feature, it's coming from the same patient at the CR9528. So the model we just looked at before. You will find the same information, examples of tumor growth curves again. And obviously, also we have tested for that specific model the same kind of standard of care. So we have compared vehicle with the sotorasib treatment and adagrasib as well in green. And as you can see, this is very interesting. We still have some degree of response with those KRAS G12C inhibitors. And what is interesting to note is that these patients here, when it has been taken, the samples has been taken later during the treatment of the disease. And you can see here after the third investigation that we had with the CR9528 PDX, at this stage of the disease, we have taken the PDX and generated the PDX later after the patient has received the fourth line of treatment. So it was so some G12C inhibitors, but at that time, the patient became resistant to the treatment. And we have tried to see how it responds to the sotorasib and adagrasib and compare that to what happened compared to the original CR9528 when the patient was still responsive. And you can see here that we have still some degree of response, as I said, but it's less. If we compare side by side, if you look at day 90 here, or day 100, so 100 days after the tumor cell implantation, you can see that the tumor growth and the tumor is above 1,000 cubic millimeter here. Same for adagrasib here. After 19 days, it's on average close to 2,000 cubic millimeter. And if you compare that to the PDX generated from the patient when it was still sensitive, after 100 days, the tumors were still below 500 cubic millimeter. So it's clear that in a way, the PDX that we have generated from the same patient is also more resistant to the treatment to KRAS G12C inhibitor compared to the original PDX samples we have taken earlier during the disease course. So that's really interesting data, and you can use those models to, for example, test a new KRAS inhibitor, or if you are interested in specifically KRAS resistant models, you can also compare side by side, for example, the response of your treatment before and after the patient has become resistant. So it's something really interesting, and I wanted to show you that and which kind of data you can see on the database and how you can use it to find interesting models. Obviously, we don't have that kind of historical data for all the 2,000 and so on, also PDX models. But if you are interested in specific standard of care resistant models, please feel free to reach out to us. We can also help you to generate some lists. I mentioned earlier that we have genomic data for the different models, and that's something that can be accessed here at the top of the screen. You can see we have the multi-omics section, and you can have access to gene expression data. We have RNA-seq data. You also have some copy number data with exome sequencing. We have mutation data as well available. We can access gene fusion, HLA, all that. So you can click here, for example, if we go to RNA-seq data, you can click here, type any gene of interest in the in this box, and then on the left select the models for which you want to have the expression data. So you can do that, and for example, if you're interested in KRAS expression in all colorectal cancer model, we have more than 400 of those. You can click here, you select all the colorectal, you click search, and you have this graph here with the RNA-seq data expressed in log2 FPKM. And you have the table below that you can download in an Excel file if you want to have those data and play with it. I just type one gene, but you can, add other genes, for example, from the KRAS pathways and the RAF pathway, for example. So if I tap that and add more genes, you will have access to that graph here, but also to all the different genes in a single table. So on the bottom, I click all to have all the models in a single table. So it's a lot of data here, and you can download that at the top in an Excel file if you click there. If you want to have more than gene expression in a single file, that's possible. We also have a search function in which you can have more than gene expression separated with copy number mutation. If I take the same example again, if I go with KRAS, for example, I'm interested in KRAS, BRAF, RAF1, sorry. And you want to have the gene expression, but you're also interested in copy number and mutation. And you want to have what is predicted to be driver mutation for those specific genes. You can click that. Again, you go to colorectal cancer, you click, and you select all 430 models, and you click search on the top right over there. It would take some time to get all the information, but once it's over there, you have all the information in a single table. You here have the KRAS gene expression, RAF1, KRAS, also copy number, and then on the right you also have all the mutation data in a single table. If you want to download everything in a single table, please make sure that you select all models here in the same table. Otherwise, it will just download the 10 models that is in the first page and not all of them. And then you can click on download again just there if you want the extra file. Actually, we can check what the models we just had a look before, like the TR9528, to double check if we have the G12C mutation that is supposed to be in the patient originally. So I search the specific model over there, and you can see that obviously in our genome sequencing data, we have found that we have found the same G12C mutation here. So we have validated that the G12C mutation is maintained between the patient and the PDX that we have generated as well. And then at the bottom over there, you have more detail about the specific mutation. So you have the source, the methods that are used to generate those mutation data. So we have data from RNA and also from DNA. So we have exome sequencing and RNA sequencing data for mutation. And then you have more information on the CDS change, mutation type, the amino acid change, the mutation frequency, if you want to know if the mutation is homozygous or heterozygous, the known variant over there, and so on. So that can be used if you are interested in specific mutation to see and to better select your models. And all of that, again, if you want to have all the mutation details, you click all to have everything in a single table, and then you can download the data over there. Okay. I also wanted to mention that on top of the classical genome sequencing data that we have, RNA-seq and so on, we also have proteomic data for some of the models. So not for genomic data like RNA-seq, exome sequencing, we have that for almost all the models, all the PDX models. But for protein expression, we also have some models that we have done. We have that we have sequenced. We have like close to 200 of them. And that can be used as well if we take the same example for the RAS and the KRAS kind of models, if you want to, for example, look at protein degraders and see how those proteins are expressed in specific models. I can take example again of KRAS, or other proteins like MEK. Sorry. Oh, SHP2, for example, and also the ERK protein. And then you can, for example, take again the same the colorectal. If we take the same example, you're interested in colorectal cancer. And then you have some data on protein expression. So you can compare the level of expression between different models and see which models express more or less of the target protein before you select that for a potential efficacy study. And those data can also be downloaded here in a single table. And you always have this part where you can download the Excel file. All right. So I think that's it for the UBase. We have also other databases for the other platforms that we offer. Now I think I will switch to the organoids. So we have a platform of organoids, either derived from PDX or directly derived from patients. And the same kind of information that you have presented here can be found in another part of the database. So on the top right, you have access to more databases. And the database for organoids is called organoid base. So if we go over there, you can see that we have access to those models. So this is the same kind of information you can find on the organoid base on all databases. As you can see on the left, you also have the search function. So the different kind of filters. If you're only interested in models for which we have standard of care in vitro, you can also use those filters to select those models. And you have the option to look at specific standard of care. Again, if you do not find the drugs you are interested in this section in the database, feel free to reach out to us. We might be able to share more information with you. We have two kinds of PDX. We have organoids derived from PDX. This is called PDXO, and we have organoids derived directly from patients. It's called PDO, for patient-derived organoids. So if you're interested only in some models, you can reduce the list to only PDO or PDXO. And again, you have where those models are available, either in China for CBCN, BSD for San Diego, and TBNL for the Netherlands, because we are doing a lot of those organoid work also in the Netherlands. And on the left, like for the PDX, you have the detail of all the models available. We have more than 400 PDXO of more than 13 close to 30 indication, I think. And then we have organoids derived from patients directly here. And we have, what is interesting, that for organoids directly derived from patients, for some indication, we have the cancer organoids, and we have also the corresponding organoids derived from normal and healthy tissue from the same patient. So that can be used to compare side by side the treatment on the cancer organoid and the healthy normal organoids from the same patient. So that's something that can be very powerful, and you can find those models on the left here. I will take the same examples because we have validated the models before, that was tested before. The colorectal cancer model that from the patient that has been treated with KRAS G12C inhibitor, and we have, for example, validated those models. We have derived, the organoids from those models. So the 28, 37, 47. All those 3 PDX models, we have derived the organoids, and that can be used obviously to do any kind of in vitro study. So for instance, before diving into an efficacy study in vivo, you can also test your compound in the corresponding organoids from the patient that has been treated with KRAS G12C inhibitors before it has been resistant or after. So that's something that is really powerful, and it will allow me to present you this part. So the summary is the same. You have information on the organoids, the name, the subtype, here is adenocarcinoma. You have the growth. So the growth speed, some models are more or less fast to grow. So you have slow, medium, and fast-growing models that can take up to 10 weeks to grow. So it's some information you have over there. You have the origin of the organoids. So it's an organoid derived from PDX from Western origin, as we have seen before. And then you can also find, again, the same information about the patient, about the pathologic diagnosis and QC as well, and treatment history as well. I think I will just take another example to show you the other kind of information you can find. Selected one breast cancer model that can be, which we have more information and more experience with. So as you can see here, we have, again, the same kind of information about the patient. For this breast cancer, you also have some IHC data, for example. It HER2 1+ positive by IHC, but it's ER negative and progesterone receptor negative as well. You can find some pictures of the organoid here in a well. And when we have some standard of care that we have tested in vitro, you can find the results on IC50 data there. For that model, for example, we have tested cisplatin, doctorubicin, paclitaxel, staurosporin, and tamoxifen, and you can find all the IC50 data after 120 hours of treatment. And all of the IC50 data are found in this table below. So you have the IC50, max inhibition, cancer type, and the assay type. So for example, to generate those IC50, we have used a CTG assay. And then you have some pathology picture of the organoid here in addition. Like the PDX, we have genomic data for almost all the models, PDXO and PDO, and this is accessible on the top here. You have the gene section. You have, again, access to RNA-seq data, copy number, mutation, gene fusion, and HLA as well. For organoids, we do not have the proteomic data yet, but all of the other genomic data can be found and looked at in the search function as well. So you can individually look, if you are only interested in the gene expression, you can directly go to the RNA-seq part of the database. But if you want, for example, to have a look at the gene expression, mutation, copy number, and I will take the same example again, use those models, colorectal cancer models. So the three, let's take two of the models, the 28 and 47, before and after the patient has become resistant to G12C treatment. And let's check several genes here, BRAF, so BRAF. And we can have a look at the genomic data. And you can see here, we double check the G12C mutation. We find the same G12C mutation in both organoids models. So we maintain the mutation between the organoids and the PDX. So that's something that we almost always see for us, very important driver mutations. We maintain the very specific driver mutation between the organoids and the corresponding PDX. So that's something really robust if you want to pre-test something in vitro before going for in vivo testing. And again, you have the same table with the gene expression, copy numbers for the different genes that you selected, and that can be downloaded in a single table. And you can have all the mutation detail in the table below as well that can be downloaded as well. I think that's it for the organoid base part. Again, feel free to reach out to us if you need to generate a comprehensive list of models. It's a good tool if you want to have a first look at specific models, if you have trouble to find the relevant information, or if you have a lot of different parameters you want to analyze and the database is not enough or is not really convenient for that, feel free to reach out to us. We can help you to generate some comprehensive Excel file with a lot of genomic data together with subtypes, PDO, PDXO, PDX, all of that together. So it can be helpful to reach out to us if you need something more complex. On top of organoids, we have another part for the CDX, so the cell lines, human cell lines. And this part of the database, you can go on the top right again on more databases and go to Xenobase. And this part is dedicated to our human cell lines. When you are landing on the page, you have access to all the in vitro data we have generated with the cell lines. On the left end of the screen, you can see the number of cell lines we have available. We have more than 800 of them that you might be able to use. We have tested some standard of care for some of the cell lines. If again, you can use the filters on the top if you are interested in specific drugs. And if you don't find it, feel free to reach out to us. We can help you to generate more data and provide you with more data potentially. We have this very interesting OmniScreen filter as well. OmniScreen is one of the screens we run quarterly. It's a screen in which we have a maximum of like 520 cell lines included, and you can test up to those 520 cell lines within 3 months, and any kind of number of compounds, so 1, 2, 3 compounds in this OmniScreen. And within 3 months, you will have the results of IT50 with all those cell lines. So you can go up to 520 cell lines, but you can also have as low as 50 models for the screen. And if you want to have a look at the cells that are included in those OmniScreen, you can click the filter and you can see all the 520 cell lines included so far of a lot of different indications, as you can see. Yeah, so if I stay there and select one specific cell line, for example, let's have a look at skin. Sorry. Let's have a look at this skin cell line here, A431. And on the right end of the screen, you will have some more information, like PDX, like organoids on the model. So here you have the cell lines, you have the species of cell line, the cancer type, the category, the subtype, yes, epidermoid carcinoma, histopathology, and also the special feature of the in vivo models if we have validated the model in vivo. And I will present that a bit later. As you can see here, we have the standard growth curves in vitro when we use different number of cells. So you have the doubling time of the cell lines for this model, and we have the information for a lot of cell lines in almost all cell lines from our OmniScreen at least. And then at the bottom you have pictures of the cell line when it grows at different percentage of confluency, and we have some example of standard of care. For this specific cell line, we have tested cisplatin, afatinib, gefitinib, gemcitabine, and icotinib, for example. And you can see how resistant or not this cell line is to those treatments. And you have all the, like for the organoids, you have the information available in the table, and you can use a search function if you are interested in a specific drug. So that's the kind of information you have for the cell lines. And for some of them, we have validated the growth in vivo as well, so you can also test it in vivo. This is available here. You can have a look at the models and tumor growth, like for the PDX, if you click in vivo. So if you go to the in vivo section, you can see on the left hand the number of models we have validated in vivo, close to 300 of a lot of different indications. I will go and the same example. A431 of skin cancer. Like the PDX, you have some information on the cell line, and you see that this model in particular can present some ulceration when we grow the tumor in vivo. You have the body weight curves. You have examples of a lot of different experiments. Here we have 22 experiments for which you have access to. You have the body weight loss, but if you click over there, you also have the tumor growth curves. You can have a look at the different experiments, how reproducible is the tumor growth in between different experiments, how fast the model is growing as well. So that's something that can be very variable. And at the bottom, we also have the standard of care. As you can see here, we have tested a lot of standard of care. If I go back to the in vitro data, we have, if you look at this curve, which is the afatinib, actually. This is a drug for which we have seen a good efficacy or the best efficacy in vitro. And for example, we can compare what happened in vivo when we use the same drug. So if I select specifically a drug, if I go to afatinib, and I will select again cell line, it will reduce the list of standard of care to only afatinib over there. If you go to standard of care tumor growth, you can see the kind of response. And here the number is the number of the experiment. So if you want to have a look only at one experiment, you can unselect the other curve, so it's more clear. you can see that in the first experiment, the treatment with afatinib is really potent, and there's a almost complete tumor growth inhibition over there. And if we look at different experiments, we see the same kind of results over here. So it's really interesting, and it correlates with what we have seen in vitro as well. Like the PDX, like the organoids, we have a lot of genomic data for a lot of the cell lines available. This can be checked at the top here. We have the gene section in which you have the RNA-seq data. We don't have the protein expression data for the PDX yet, but you have access to copy number with exome sequencing, mutation data, HLA. So you can, again, look at specifically RNA-seq if you go to this section, but if you want to look at the same time gene expression, copy number, mutation, or mutation, gene expression, you can select only some of those information, add your gene of interest in the box, select the model that you are interested in or indication of interest, and then generate the same file and the same kind of information we have seen for organoid base and also for U base for the PDX. So I think I'm over with the database, and I will finish with the last part of the database. It's called new base, and this is a section in which we have all the syngeneic models, mouse-related models. A lot of that is used for immuno-oncology studies. When you are landing on the page, in the first page, you have access to the mu prime, which corresponds to the mouse tumor homograft. For the mouse tumor homograft, this is models that has been generated in GEM or carcinogen-induced tumor. So those primary tumor has been generated either in mice that have specific mutation and that just spontaneously develop the tumors, or that has been generated with carcinogens. We are taking the primary tumor and then we reimplant those tumor in other animals subcutaneously or orthotopically. And then we can study the different kind of treatments. If you're interested in specific driver mutation, some of those models can be interesting, and compared to cell line, that can be also more physiologically relevant because it's not like a cell line that is clonal, that has clonal expansion. It really has been generated with carcinogen or with specific driver mutation, but then the tumor has been developed and spontaneously developed in the mice, and it's not a clonal expansion of one cell line. You have on the left there the different indication and models, like for all the other parts of the database. You can see we have a lot of indications available. Here on the right part, you have the detail on the model. So the kind of mutation that has been used to generate the tumor homograft, the mouse strain, the biopsy site from the primary tumor. If there's special feature in that one, we know that there can be some iteration. If we have the immune profiling, so in that case, yes, we have the immune profiling. It means that we have checked the kind of immune cell infiltration we have in the tumor and also in the periphery of the animals when we implant the tumor. So that can be very interesting, and I will show you the data just at the bottom of the page. And then you have detail about the model itself, about the GEM mice or the carcinogen-induced model. And then you have some reference that can be downloaded here in the PDF if this is of interest. And again, some reference of tumor microarray, because we also have tumor microarray for those tumor homografts. Same as all the other sections, you can have access to body weight curves, tumor growth curves, and I will try to standard of care as well, obviously. And you can see we have tested chemotherapies with that specific breast tumor homograft. We have also tested some checkpoint inhibitor for those models, and that can be checked over there in these graphs. You also have access to, in that case, the immunophenotyping. So if I click over there, you see this graph where you have the percentage of different immune cell population in the lymph nodes here. Just to mention, these models has been implanted orthotopically, so this is the data from orthotopically implanted mice. If you go on the right, you have also data on the peripheral blood for B cells, T cells, Tregs, NKT, NK cells, and also macrophages and myeloid cells. And you have the data on the spleen and in the tumor as well. So you can have an idea of which kind of immune cell population are infiltrating the tumor with that specific model. And then at the bottom, you have some pathology pictures of the tumor homografts of the mu prime. Okay, and then in another part of the database, if you click on models on the top, if you want to have access to the syngeneic, more classical standard cell lines, mouse cell lines, it's just over there. You have access to more than 50 cell lines, mouse cell lines. This is the same structure of the database. You have the filters over there. One of the filters that is really interesting, I want to mention, is a mute screen. Like for the Omni screen for cell lines, we also have a screen in vivo for the syngeneic models. So in total, we have a maximum of 12 models in that screen that are in quarterly as well, and you can test any kind of drug against all 12 models at the same time, and you will get the data within 3 months. If I take a specific model from this new screen, like M638, a colorectal cancer model, and see how it looks like, same kind of structure, you have the name of the model, description of the model, if it's available in China, here in the UK, and in San Diego as well. So all 3 locations, we can run the model. And then you have at the bottom examples of tumor growth curves. As you can see here, we have a lot of experience with those syngeneic models, like more than close to more than 100 experiments, and it gives you an idea of how fast the model is growing and how reproducible is the tumor growth in between different experiments. And then you have different standard of care. We have tested obviously a lot of the checkpoint inhibitors like PD-1, PD-L1, CTLA-4. So we can have a look at the tumor growth curves here, and we can share more dedicated data pack as well with you. If you reach out to us, we can also provide you with more data potentially. So you have the tumor growth curves, and if you are interested only on PD-1, you can again select some tumor growth curves. If you want more comprehensive data set in slides, we can also provide that to you. Sorry. I will finish with those syngeneic models, talking about the genomic data. Again, we have the same genomic data like RNA-seq, copy number with exome sequencing, mutation and gene fusion data, and we have protein expression for the models as well. I will go to RNA-seq for example, and give you an example. You see on the left, we have the data for the mouse cell lines. So it will be the data, RNA-seq data in the cell lines in vitro. And but if you go at the bottom, you also have the data in what we call syngeneic models. It would correspond to the tumor. So we have the RNA-seq data for the cell line and the tumor in vivo. And obviously, we also have the genomic data for the mu prime. So if you are interested in, let's say, in anti-PD-L1 treatment, so I will type the code for the PD-L1 gene, and you want to see how it is in the syngeneic models in the tumor, and you want to have a look. It can give you an idea of how PD-1 is expressed at the RNA level and compare all the different models between them. So that can be done with any kind of target and can be used to select a model based on your specific target, obviously, before running an efficacy study. And then for the syngeneic model specifically, we also have proteomics data, and that can be accessed over there. If we go to the gene section, you go to protein expression. And then if we type the same gene, if we select a couple of models like the lung model and the breast model, for example, and then you see here the comparison in terms of protein expression, and it looks like PD-L1 is expressed over there, and that LLC, the lung model, is expressing slightly more PD-L1 compared to EMT6. So that's also a good tool if you want to compare the protein expression and compare that in between different models to better select the one that is interesting for your own targets and your own therapy. And I will mention real briefly, just to finish the 2 last sections, we have gem mice, so genetically engineered mice, and that specifically DTR animals over there. Those DTR animals are mice that have a DTR expressed under a specific promoter to deplete specific immune cell populations. So we can have models to deplete CD8 T cells, CD4, the Tregs here, B cells, myeloid cells as well, macrophages. So this is really interesting if you want to do some mode of action kind of study to better understand how your drug is working. And you can download some additional information on those DTR mice in a PDF, and you have more examples in those slides, and all of that can be downloaded on the database. And last section I will present today is a UGEM. So this again, genetically engineered mice to express a specific human target. So that's a very, very powerful platform that can be used to study your human antibodies or targeting drug against a specific target in an immunocompetent animals. We have here a list of the mice for which we have the humanized version. So you see you have a humanized target for a lot of molecules. I will take, for example, the UGEM for PD-1. So if you have a human antibody targeting PD-1, we have a UGEM for a mouse model expressing a human PD-1. Like for the other gems, you have more data that you can download in a slide deck. And what is interesting is that we can use those models, those UGEM, together with what we call U-cell, and you can find the information over there. So U-cells are, again, cell lines that express a specific human target. So for instance, if you want to have both human PD-1 and PD-L1 expressed on the tumor, we can use a specific tumor cell line expressing PD-L1, like MC38, for example. We have the MC38 expressing human PD-L1, and we can implant the MC38 human PD-L1 in the human PD-1 UGEM to have like a complete receptor ligand humanized crosstalk. Just to mention, we also have double knock-in mice. So if you want, we have the PD-1, PD-L1 mice. That can be very interesting, but you have the full list over there, and you can download more data in a PDF format. I just want to mention as well that we have a list in the database here in the new base, but we have access and we're working with different providers, so we might have access to a lot of different other UGEMs for other different targets. There's different level of characterization that has been made, but if you're interested in a very specific human target and you want to study that in immunocompetent animals, feel free to reach out to us. We can check if we can access a specific UGEM that you do not find on the database right now. With that, I think I'm at the end. I thank you for your attention, and I'm ready to take any question you might have on the database. I know it was a lot of information. The goal was to review and to go through a bit all the parts of the database, but if you have any comments or information, feel free to ask them now. Thank you.